Good evening, everybody. Much love. Bringing you guys another video. It's kind of a little bit strange, but I wanted to showcase the research findings that I came across last night. So I'll go over to my Twitter so I can show you guys this thread I developed, or am developing still. And this is, oh, that's just shameless. This treacherousness. And so this is what I was talking about earlier in the chat. This company right here, which is formerly this company right here, Synthetic Genomics. I'm going to read briefly from some of these articles I posted into my thread here, and show you guys a little timeline, a strange timeline for this. So basically this is an article here that's very telling, and it's very old. It's not a new article, but it reads similar to the stuff that we're all talking about right now. So I'll just so JCVI is kind of like a focus of my research, and I'm just gonna go through this. So the Synthetic Genomics Incorporated is a company, SGI, and people sort of barely know about that, but they definitely barely don't know about this one, Synthetic Genomics Vaccines Incorporated, SGVI, if you see right there. So I'll read a little bit. So JCVI have formed a new company, Synthetic Genomics Vaccines Incorporated, to develop next-generation vaccines based on synthetic genome technology. The new firm has already inked a 3-year alliance with Novartis to develop a bank of synthetic seed viruses as a rapid response technology for the production of annual or pandemic flu vaccines. The collaboration will be supported by a U.S. Biomedical Advanced Research and Development Authority (BARDA) grant. SGVI will exploit the JCVI's expertise in genomic sequencing and synthetic genomes. Yada, 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 and it goes through a bunch of that, talking about how they make the actual, see, and it even goes into this. They tell you in here, they tell you if you read through this, but anyways, back to the thread, that's just showing you what the synthetic genomics is. Again, fair use. This is my science thread, and this is all open source. These are, so this is a original Cynthia component. So part of the original was a bacteria found that thrives in the ocean floor, and if you read the article or the wiki, you can get into understanding what I'm saying when with that. It kind of relates to the discussion we had earlier as well. And I have two threads. One of them is like the live stream thread with all the live streams, what I'm talking about. And then this is actually the header thread of the one that I'm going to present, that the information I'm presenting right now. And this article actually is very telling as well. If you look into this article, it's a little bit newer. It's a 2015. And if you read it, it reads like they're debate. I'll read a little bit of this. Fair use. It reads like this is like telling you what they're gonna do to you, basically. So it says your entire genome will be sequenced with extraordinary resolution and accuracy. Your body will be scanned in fine three-dimensional detail. Thousands of compounds in your blood will be measured. Even the microbes that live inside you will be surveyed. You will get a custom-made iPad app to navigate data about yourself. Also, your wallet will be at least $25,000 lighter. Venter, who came to the world's attention in the 90s when he led a campaign to produce the first draft human genome, launched Health Nucleus last month as a new part of his new company, Human Longevity, HLI. He has made clear that his aim is just as lofty as when he and his team sequenced the human genome, get this, or built a flu vaccine from a genetic sequence delivered to them over the internet. And then it just goes into all this. And it tells you what it is and who they are. And there's like a video or two in here that's very telling. Like this one, for example, is just
spectrum. So if we can,
they talk about
life-saving technology. Human Longevity Inc. is the largest genome sequencing facility in the world. Here,
so yeah, they get into all, like they show, they're like selling this procedure that seems to me like set for the future anyways. But so that's the head of the thread, and then I have this article. You have to have a subscription to even look at it, but it tells you that they're behind making the stuff. This is an article from today that's like really shameless, so I just put it this way. Basically, this is the YouTube channel that I originally learned about all this stuff back in 2010, so I knew they were going to develop like all this stuff that we're facing now that they're using. And okay, so this is I think I showed this article. This one is the guy's actual company, and this is the actual Corona V research actual, and then these are the actual research team that I've never heard anybody talk about. Like, we've posted it a little bit before. I've showed this a little bit before. But anyways, yeah, if you go in here and look in here, all of these are in here. Like, this is their science technology. And so what I found was that what this tweet is about is this company right here, the one I found, formerly Synthetic Genomics. And if you go into their website, it's very strange. I'll show you guys this.
Some farms grow food. This one grows fuel. ExxonMobil is growing algae for biofuels that could one day power planes, propel ships, and fuel trucks, and cut their greenhouse gas emissions in half. Algae. Its potential just keeps growing.
Viridos. applied knowledge to address unsolved challenges in pharmaceuticals, energy, and life sciences. Obviously, this is like some front company that they just made because they don't want to have the association with the genomics necessarily, but they're like exactly doing all of this stuff. And then, okay, here, so this is what I was talking about, the strange timeline. It says in here, in this site, we've been innovating on behalf of the planet and its residents since 2005. Since our founding, we've pioneered genomic innovations and have been incorporated into new technologies, spun off into companies across multiple industries, and codified in more than 100 patents. And then June 2007, first genome transplanted, a key proof of principle in synthetic biology. Our transplanting of the first genome was an important step towards realizing the goal of creating a synthetic organism that can help solve global health and sustainability challenges. January 2008, first bacterial genome sequence, the synthetic genome, the largest man-made DNA structure created, was regarded as a striking technical accomplishment and formed the groundwork for our synthetic biology platform. Isothermal DNA assembly, the assembly method first published in Nature Methods, can be used to seamlessly construct synthetic and natural genes, genetic pathways, and entire genomes and functions as a useful molecular engineering tool. April 2009. July 2010, we developed the first self-replicating synthetic cell, a bacterial cell that was designed in the computer and brought to life through chemical synthesis without using any elements of natural DNA. This enables the development of many important cell-based applications, including vaccines, pharmaceuticals, biofuels, and food products. So, to be continued, I clicked more. May 2013, synthetic flu vaccine. Response to a global health threat posed by the H7N9 bird flu, we developed a synthetic approach that rapidly generated a vaccine seed from downloadable sequence. May 2014, automated DNA, RNA, and protein production. Veridos developed a cloning module, a new capability that automates and expedites DNA cloning, a procedure used in nearly every academic and commercial molecular biology laboratory worldwide. Based on this innovation, we developed an automated manufacturing process that significantly reduced the time it takes to create DNA constructs for therapeutic research and development programs, clearing the path for a wide range of research applications. At the core of this process is the BioXP, the world's first benchtop DNA printer and automated instrument that enables biotechnology, pharmaceutical, academic, or governmental laboratories to create DNA from electronically transmitted sequence data. We successfully spun off this technology in a new company in 2019, now called Codex DNA. September 2014, emerging phage. We developed supercharged phages and type of virus to combat antibiotic-resistant strains of pseudomonas and a bug that causes skin infection, sepsis, and potentially fatal pneumonia. Phage therapy has a potential to treat multi-drug resistant bacterial infections. Based on our engineering phage, we developed a proprietary bacteriophage engineered platform to address the inherent limitations of natural phage as a therapeutic. Armata Pharmaceuticals is now developing the Viridos IP to generate clinical candidates in bacteriophage therapies. March 2015, lipid switch. Viridos discovered the genetic switch that enables the concurrent growth and increased production of lipids. March 2016, first minimal cell. We built the first minimal synthetic bacterial cell. This is the smallest genome of any organism that can be grown in laboratory media. This feat led to new tools and semi-automated processes for whole genome synthesis, aided in more efficient development of biologic drugs. June 2017, algae biofuel breakdown. Together with ExxonMobil, we modified an algae strain that more than doubled its oil content without inhibiting the strain's growth. The results were published in peer-reviewed journal Nature Biotechnology. February 2018, engineered pig cells growing organs for transplantation. In partnership with United Therapeutics, Veridos developed the capabilities to engineer pig cells with the goal of generating humanized pig organs for transplantation. Based on the IP we developed and its preclinical progress, United Therapeutics assumed full control of the program with the goal to generate xenotransplants for the 1 million plus people in the U.S. alone who have end-stage organ disease. July 2018, trait stacking and gene knockout, published in the Proceedings with the Nation Academy of Sciences, PNAS. We successfully stacked 7 different genes in a microalgae's genome, as well as knocked out redundant genes. Ultimately, this allows for single strain to express multiple desirable traits simultaneously. October 2018, nutritional proteins and oils spin-off. Acknowledging the global need for more sustainable vegetarian food protein sources, our discovery and improvement of algae strains that ferment sugars into high-quality nutritional proteins and oils provided a solution. Based on this work, Small Foods, a recently launched company, developed food, proteins, and oils from non-engineered algal strains developed by Viridos. March 2019, our Algae Biofuels collaboration goes outdoors. Our long-standing collaboration with ExxonMobil took a deep leap forward with the implementation of California Advanced Algae Facility, CAAF, where we are piloting the next generation of our joint algae biofuels program. May 2019, self-replicating RNA medicines, innovative vaccine and therapeutics. We developed a proprietary RNA replication platform to selectively stimulate the immune system and deliver targeted therapies produced within a person's own body. The RNA platform overcomes common shortcomings of traditional replicon and messenger RNA, mRNA technologies, enabling a new class of vaccines and combination therapies to combat cancer and infectious disease. The platform has been successfully partnered and with leading human and health companies. August 2020, outdoor algal bio performance reaches new high. Viridis achieve outdoor algal bio oil productivity of 5 grams M2 per day, an industrial relevant scale at the California Advanced Algal Facility, using our best performing proprietary microalgal strains. At this, substantially higher than what has been reported in the field at key milestones towards our goal of deploying microalgae to produce low carbon intensity fuels at scale. Veridos secures an additional EPA approval for outdoor cultivation. May 2021, Viridos obtained additional EPA approvals to conduct outdoor cultivation of our lead algal biofuel strains. By granting the ability to perform extensive environmental monitoring, we can transfer to our most productive strains from the lab to a real-world setting. This real-world performance accelerates a virtuous design, build, test cycle of continuous improvement in strain productivity and robustness, and provides evidence that our strains are not harmful to the surrounding environment. Anyways, I don't want to make this video too long. There's probably a lot more in there. Wanted to show the strange timeline. I originally learned of all this information back in 2010 from WVP Radio. If you go to this video in this thread, the actual video
Thank you for joining us on a special edition
is called "It's Not Wise to Fool Mother Nature," and I'll leave it there. Fair use, peace and love. Bless.